Okay, so my name is Dr. Nuytemans. I'm originally from Belgium, actually, and I moved to the US like 12 years ago. I work at the University of Miami at the John P. Hussman Institute for Human Genetics. That's a mouthful, but basically we do genetic research for any kind of human genetic disorders. Yep, yep. Let's see. Okay. So I will talk a little bit about what is genetics, and then I'll talk a bit more specifically what we know of genetics in dementia in general, so both for Alzheimer's disease as well as some of the other related dementias, and then what we're doing currently at the Hussman Institute for Human Genomics. So I want to ask a very general question. Who's heard of genetics or knows a little bit about genetics? Okay, quite a lot of hands. And now honestly, how many of you actually understand what it is? Yeah, I'd be even hesitant to put up my hand. I've been doing this for 15 years, and the more we learn, the more I know. I don't know anything, so it's a work in progress. Okay. So, very basic, our genome exists of 4 bases, right? The letters A, T, C, and G, and in however combination you have those letters in order, they mean different things. They code for different proteins. And our genome is pretty big, so it's very, very long. So we actually have 3.2 billion of these letters per cell. So that means it's actually over 6,500 phone books per cell that information is contained in. That's higher than some of the biggest buildings we have here in the US, right? So how does that actually work in your cell? So your DNA is organized in this double helix form. I'm sure you've seen that shape in many genetic information websites or things like that. So our information is in those helixes that then get folded up and folded up and folded up until we get these, give me this side, until we get these chromosomes, right? You might have seen some pictures of that as well, those little bundle up chromosomes. So a lot of information get condensed in a very, very, very small shape. So each of these chromosomes has functional units, what we call genes. You've heard of that before. Dr. Galvin has mentioned some gene names before, right? And when you do genetic testing, we test only small parts of your human genome. And each copy of your genome has over 22,000 genes. So that's a lot of genes. There's a lot of information in each of your cells, and each of these genes codes for a different protein. Now, how does that work in your cells, right? Because you have all kinds of different cells, right? Skin cells, brain cells, kidney cells, retinal cells, right? Do they all use those 22,000 genes? No. So there is a very specific usage of each of these genes per cell. And I'm going to try in the rest of my talk to sort of compare it, make an analogy to music instruments and musical concerts that you go to, right? So you can go to a classical concert where we only use the flutes, the violins, the cellos, and things like that. Or you can go to a rock concert where we just use the electrical guitar, a drum set, and so on, right? So each cell uses different proteins and different genes to get to the function that they need to go to. Now, each of our genomes has variation, and some of that variation is completely normal, and it actually makes us different between us. Because even the Queen and Will Smith, right? They are 99.9% identical, genomic-wise. There's only a 0.1% that makes us different from each other. But remember, our genome is 3.2 billion bases, so 0.1% is still a few 100,000, right? So there is a good amount of variability that we can have. So when we go back to our music analogy, you can think about it as even the same music sheet can be played by different people, and it comes out as the same sort of symphony, but it sounds maybe slightly different if different people play it, right? So it's eventually the same thing, but it comes out maybe slightly different. And that's our normal variation. Now when you have mutations, right, maybe the A at a certain position gives you the normal protein, where the orange protein can interact with the blue proteins and do its normal function. But if you have a mutation, so for example, if you have the C, let me see, the C here, your protein doesn't fold correctly and it becomes abnormal, and it can no longer interact with the blue protein. So what could be what could we mean by abnormal? So you can have a loss of function. And again, to our music analogy, you can be like, okay, I'm having a classical concert, but all my violinists didn't show up. So you're missing a pretty integral part of your actual symphony, right? Or the other way around, having that mutation or the abnormal gives you a gain of function. So now suddenly an electrical guitar player shows up at your classical symphony. That's going to throw things off, right? He's going to overpower everybody else that's there. The conductor doesn't know what to do with that, so everything gets a little out of sync. Or maybe it's not an extra instrument that gets added, but maybe suddenly there's too many of the flutes, and they also overpower some of your violin players or something like that, right? So there's a imbalance between the genes that should be there and how much they're being used to make proteins. And some of these changes, right, might not be that detrimental, right? They change things up a little bit, they mess things up a little bit, but it's not that bad. Again, in our music analogy, your conductor can sort of tell that musician, play a little bit softer so you're not overpowering as much, right? So the cell also has sort of these compensation mechanisms to try to fix it a little bit. But in the long run, your cells get tired, your conductor gets tired, and things start getting out of form again, right? That's why it happens sort of with late onset disorders. The dysfunction might be happening many, many years ago already, but you don't see symptoms yet, it's not as bad yet, but over time, your cells get tired, they then start to die and actually show your phenotype, right? So that's sort of the same idea. It's hard to keep talking into the mic while also trying to point. Okay. So what kind of different genetics diseases do we have? So we can have a single gene disease or a monogenic disease, which means that the mutation that you have in one gene is sufficient to cause disease, which also means that if you transmit that gene with the mutation to your child, that they will also probably present the disease. So by itself, it's sufficient to get the cell completely out of function. So that's, again, the analogy, that's the electrical guitar that shows up. It throws everything off. The other option is a complex disease. So it's not just one instrument that shows up or one instrument that's completely missing. It's everybody's like a little bit off sync. There's multiple small risk factors that by themselves are not enough to cause disease. But if you're unfortunate and you have a combination of many of those things, you might present with disease. And in this case, in these complex disorders, we sometimes also have the influence of our environment. So those are the factors that Dr. Galvin actually talked about, your resilience factors. What's your education? Where do you live? What has been your job? Like, so those are things that also influence. Again, in my music analogy, I sort of put here, it's the music hall, right? Depending on whether you play here a rock concert or in an actual big arena, it's gonna sound different, it's gonna mess things up a little bit depending on where you are. And I like to sort of liken this to you're playing in a symphony, but maybe some of your players are not as good as they're supposed to be. So it still works, right? Like one player is slightly off, but your conductor can match with it. But if you have a bad player, if you have an instrument that's not there, and you play in a music hall that's not really meant for that kind of music, that all starts adding up. And eventually you're gonna get to a concert that's not really that well or that good. Your listeners sort of get fed up with it and they leave. It's basically your cells are dying, right? So it is the addition of different factors that are only slightly off that together are enough to cause disease. Okay, so now why do we do genetics, right? That's why am I here, and why is Dr. Galvin also interested in genetics, is because it tells us a lot about the disease. So he talked a little bit about diagnosis, right? If we know genes that are involved with certain disorders, if we can identify a variant of that in that gene in an individual, then we can sort of be more confident saying like, yes, it is this clinical phenotype that this person has, and do more targeted treatments. For some variants, we can also sort of use that information to predict how the disease will evolve. We can also if you have that variant in people that don't have any symptoms right now, we can sort of predict, like, will they have a higher chance of developing disease or not? From a more scientific standpoint, which is where I'm at, it helps us understand what's actually happening biologically in these disorders. So what is throwing it off? Is there certain proteins that are messed up? Is there a problem with the mitochondria, with certain organelles in your cells? So it helps us a lot in trying to understand what's actually happening. And eventually, or ideally, we want to get to, it helps us determine drug targets. So, okay, let me see, go to the next stage. So, Dr. Galvin mentioned that a lot of the treatments so far have also been based on symptomology. They try to prevent the symptoms from happening. That doesn't help in the long run because the pathology, what's happening before that, is still present. We're not stopping disease, we're not reducing disease, we're not curing disease. We're just trying to make the patient less symptomatic. Whereas when you can target the genetic target, change the genetics, you're stopping the process a lot earlier and might be able to stop disease, maybe hopefully eventually cure the disease before it actually happens, right? So we've already seen that in the general medical field, and unfortunately we're not quite there yet for dementia research, but we've already seen that treatments that target genetic targets are twice as likely to be successful in clinical trials than any other clinical trials that are out there. So it's very encouraging for us as geneticists to keep doing this work and hopefully be able to help a lot more people in the future. Okay, so now shifting back, like what does that mean for dementias, right? What kind of genetic research do we do? So I have a little table here, which I can't see from my angle, but I'm trying to point to the right one, comparing Alzheimer's disease with frontotemporal dementia with Lewy body dementia. And I had a few people ask me there when I was standing by her brochures for a genetic study, like, what's the difference again? So most of you know Alzheimer's disease, right? You forget memory. Most of you who worked with Dr. Galvin before and have heard him talk probably know a little bit about Lewy body dementia as well, right? It's the dementias, it's a dementia phenotype combined with more of a Parkinsonian phenotype, so movement disorders. And I'm actually kind of interested in the middle one, the frontotemporal dementia, which is more sort of your interpretation of words and interpretation of social situations and how you respond to that and changes in that over time. So most of the research has been done on Alzheimer's disease because, as you can see here, it's by far the more frequent dementia that's out there, right? But we do have some preliminary research on frontotemporal dementia and a little bit less on Lewy body dementia. So Alzheimer's disease and frontotemporal dementia are very heritable, have a very high genetic background, so both in single gene disorder form and in the complex disease disorder form. So I'll go with that a little bit. So when we look at Alzheimer's disease, we have 3 genes that are the main Alzheimer's disease genes that we know of right now. So APP, PSEN1, and PSEN2. That doesn't mean anything, but there are 3 proteins that are very much involved with causing the pathological changes that Dr. Galvin talked about as well. So the amyloid plaques. All these three proteins are either the actual protein in the plaque or 2 genes that make the plaques, actually, that cause the changes in the formation that makes the plaques form. That's mostly for early onset cases, so those are the cases that are monogenic, right? 1 change is enough to cause disease. A lot more patients have a late onset Alzheimer's disease, so those are the complex disorders, right? Where all my musicians that don't really know what they're doing, they're playing together, right? There's multiple little proteins that are a little bit off. So that's my late onset disease. It's more complex, so we know a lot more genes there. And APOE is a major one, and then we have ABCA7 and over 20 more additional genes. And actually I want to talk a little bit more about APOE. So APOE is the strongest risk factor for APOE. It's not a monogenic disorder. It's not like you have a variant and you get disease, but it's pretty close out there. And it's actually important for my getting into how ancestry actually can play a role in genetics. So this is a different depiction of what I just sort of said. The 3 genes, APOE and APSN1s here. They're the monogenic forms, right? So those are the wrong instruments that show up to your classical symphony. And then the ones on the bottom here are all the genes, or a good number of the genes that we've identified that slightly increase your risk if you have a variant in that for Alzheimer's disease, but by themselves, not enough. But if you have enough of the variants together, you might actually get the phenotype as well. Let's see. Okay. So APOE is the strongest risk gene for developing Alzheimer's disease, and we've known for this gene for a long time. So this was identified in 1993 by my current boss here, Dr. Perishak-Vance. She's the director of our institute in Miami. And even though we've known about this gene for the longest time, we still don't really know what it does, which is unfortunate, but we're starting to get there to understand how it plays a role in Alzheimer's disease and how we can influence it with gene therapy to see whether we could potentially fix it. But there's 3 forms. There's a 2, a 3, and a 4. We made it easy, just give it numbers. That's easy to remember. The 3 allele doesn't really do anything. It doesn't increase your risk or decrease your risk. Number 2 decreases your risk, and number 4 is the important one. That's the one that increases your risk. As you can see on the graph here, if you have 2 copies of number 4, you have more than 80% chance of developing Alzheimer's disease by the age of 75. So that's pretty big. Now the caveat that I want to add to this talk is that this is the information that we originally got in 1993 and the years following, and most of that study was done on people from European descent. As we went on and studied more and more Alzheimer's disease patients, we started seeing some weird results in some of the other populations. So you can see here, East Asians, they have a lot higher risk. So if they have 2 copies of the APOE4, they're a lot more likely to get Alzheimer's disease early on. Whereas in African Americans or in Yorubans, which are Africans from actual Africa, their risk effect is a lot smaller. It still increases your risk for Alzheimer's disease, but it's not as strong as in Europeans, which struck us as odd, right? How do we explain that? So we've actually seen that ancestry plays a role. So I want to make sure that I make the distinction between race and ancestry. Race is generally a culturally ascribed term, right? So in ancestry is actually the one that's biological, and it's based on the history of your genetic variation. So most populations today are actually an admixture of different genetic ancestries, mostly European, African, and then Asian ancestry. And then here in the US, a lot of Amerindian as well, right? Native American. So just to illustrate what that means is here on this slide, these are 4 people that identify as African American. What does the picture look like? So how many chromosomes do we have? People who know about genetics? 22, yeah. So each line is a chromosome, and we've colored them based on what we know about the genetic ancestry in that individual. So based on the normal variation that we have in each individual and each chromosome, we can sort of deduct whether something was from an African ancestry, a European ancestry, and so on. So we can depict that for each individual. So for example, here, this individual has a lot of African background, right? Mostly yellow. This individual has about 60% European, 40% African, but then the one on the bottom here has 80% European and only 20% African. Again, they all identify as African American. They might not look any different from each other, but genetically they are different. And depending on where your gene is located, you might actually get a gene that was from European ancestry or from African ancestry. Same sort of example here for Hispanic individuals. So these are 3 individuals from the island of Puerto Rico. You can already tell by the colors, they're very different, even though they look the same, they feel the same, right? They all identify as Hispanic and being from Puerto Rico. So one is mostly European, one has a lot of Amerindian background, and one has a lot of African background. So for us as geneticists, this is important to take into account. Because when you pull it back in with the APOE gene, we've actually identified that if you are, for example, African American, your risk is lower because if your APOE4 allele is on the African background, you have a lower risk. If your APOE4 is on the European background, you have a higher risk. So if you're an admixed individual, so an individual who has contributions from different ancestries, it is very important to take into account where your gene is coming from. Which of your ancestors is it coming from? And if you want to think about it, like how does that make sense, right? Because it's the same mutation that is happening in this individual. So how does ancestry play a role? How does that influence what the effect is of this variant? Think about it as, again, sort of your musical conductor, right? You have the same musicians there, the same issue is happening, but some conductors might be better at sort of telling that musician, like, you play a little bit softer, please. And they have other ways of compensating for it, where some conductors might not be that good at it. So there's a little bit of other factors around that influence what the effect of the variant is going to be. Right? So what if all of these genes sort of taught us about what's happening in Alzheimer's disease itself? So the monogenic forms, so again, the APP and the PSEN1s, I already sort of told you that they're all playing a direct role in amyloid plaques. I hope I'm pointing at the right one. And amyloid plaques over here. So they have a direct role, and we see a very clear correlation between mutations in this gene. Yes, it's exactly what we're seeing in the brain, so yes, that makes sense. The other genes that are more the complex forms, or the bad musicians, we don't see that direct correlation between mutations in there and an actual role or a physical protein that we can see accumulating in the brain. But we sort of start seeing patterns here. These genes are involved in protein clearance. So if the final thing that's happening in Alzheimer's disease is having an accumulation of these plaques, well, if you have proteins involved that should be removing those plaques, and those are not functional anymore, that has an indirect effect on those plaques, right? Immune response, I think Dr. Galvin also mentioned that some of the brains have a clear inflammation phenotype. I pointed at the wrong one. Inflammation phenotypes, all of those little factors play a role in the eventual health of your brain. Right, so we're starting to see patterns. And then lastly, we've noticed that obviously ancestry is important, that the same variant on different backgrounds can have different effects. I didn't have any examples on these slides, but you can even have the same gene with different variants having different effects. So we know of a gene that has different mutations on a European background or an African background, and they have very different risks for Alzheimer's disease. Actually, the variant on the African background, in this case, is a higher risk than the one on the European background. So it's very much important to keep ancestry in account. And then obviously we can have different genes that are involved in different backgrounds as well, right? Going back to our frontotemporal dementia, or to our table here, where we just discussed somewhat Alzheimer's disease and what's going on. For frontotemporal disease, we're a little bit sort of lagging behind on the Alzheimer's disease field, and that's mostly because obviously Alzheimer's disease is a lot more common, and then we have our frontotemporal dementias and dementia with Lewy bodies that is a lot less common. So some of you have also asked me, what is frontotemporal dementia, right? We have two different forms. There's a behavioral form where you sort of lose interpretation of words and sentences. You can still read it, you still can come up with the word, but you don't really understand what it means anymore. Or you have changes in appetite, changes in behavior. So in a social setting, you sort of lose what a normal reaction would be, and you act inappropriately. The second form is more of a language form, and I don't know how many of you heard of Bruce Willis's diagnosis several weeks ago or 2 months ago of aphasia, right? So in that case, it's a loss of what that word means. He lost, if he could read a script but no longer understand sort of what it meant that he was reading, and had no longer the ability to repeat what was in the script. So this is a slightly more specific phenotype than that, but it's the same idea of aphasia. About 40% of frontotemporal dementia patients have a positive family history, which means that there is a strong, more monogenic form present in these individuals. Unfortunately, because it's less common in Alzheimer's disease, the research has somewhat lagged behind a little bit in terms of getting enough patients together to study this, the genetic background of it. But same for Alzheimer's disease, we know some. So we have, again, 3 major genes that we've identified. MAPT, so tau, granulin, and the C9orf72. When you see this kind of notation, it kind of indicates that we don't know what it does yet. It's chromosome 9, gene 72. So we haven't really figured out what it does yet. Generally, a gene name gives you an indication of what the protein does, so what we've learned about it. In this case, we're not quite there yet. We see people have mutations in this gene, and it seems to correlate with disease in a family, but we don't really know what it does yet. And then we have a lot of unknown, like we don't really know yet what those genes are. So very early stages here. And again, with the caveat, most of this research so far has been done in people of European descent. So what have these genes learned us? It's actually quite similar to Alzheimer's disease. So similar, we see that the genes that were identified in the monogenic forms are directly related to what we're seeing pathologically speaking. So this is kind of a colorful depiction of what we know of clinical phenotype, the pathology, I think I'm pointing to the right one, pathology, and now what we know about the genes. So we have the behavioral form, that's both green and red. So we see both tau pathology and this TAR-DDP, which is just another protein that accumulates. And you see that the tau pathology has mutations in tau, the TAR-DDP pathology has mutations in the TAR-DDP gene. So again, a very direct correlation. There's other genes that we again not really know what to do, but kind of similar to what we see for Alzheimer's disease. They're often involved in protein clearance, inflammation, immune response, and things like that. So there seems to be somewhat of a common theme across all brain disorders as to what sort of genes are involved. It's just when you start combining different factors together, different musicians together, you get one phenotype or another, right? And again, caveat, most of this done is on European descent patients. To illustrate what that means, because for Alzheimer's disease, because it's so more common, we actually have research studies in Europeans for 93,000 individuals. Those are sort of the cohorts that were data sets that we're talking about in genetic research. You can already see that even for Alzheimer's disease, the number in the other populations is a lot smaller, or other ancestries, right? 8,000, 4,500. In FTD, the biggest data set we have is 3,500 because it's so much less common than Alzheimer's disease. So we have to work harder to get our participants and get enough data together to get us to that stage where we can identify what all the genetic factors are. You can see I have a bunch of question marks for Hispanic population, African Americans, and Asians. So there's definitely a gap there that we still need to fill. Okay, but why is it important? And especially for frontotemporal dementia, actually. There are currently clinical trials underway that are specifically targeting those genes that we've identified for frontotemporal dementia. So these are the first studies that are doing actual gene therapy clinical trials, trying to prevent very, very early on, targeting the very basic cause of the disease and not some of the symptoms, right? And you see here, phase 3, it basically means they're already treating patients, right? It's no longer testing whether it's safe or not. It's actually trying to treat patients. So especially for frontotemporal dementia, we sort of skipped some of the steps that Alzheimer's disease has been doing, trying to target symptomology. For frontotemporal dementia, we skipped that and are going straight to the gene therapy, which is very exciting. Okay. Last section here, the Lewy body dementias, which on my slide put clinically. Dr. Jim Galvin is the clinical expert. You're in the best hands with him when you want to get some more information about the clinical phenotype. As I mentioned before, it's sort of more of a joint presentation of movement disorders as well as dementia symptoms. Interestingly, though, the family history here is not very common, meaning that it's very likely in more complex genetic forms. So not the wrong instruments that show up, but more of a combination of bad musicians, and it's not the right music hall, and the conductor doesn't know what it's doing. So it's sort of a combination of different things together. And again, because it's less frequent than Alzheimer's disease, it's not, the research is somewhat lagging behind or has been delayed, trying to figure out what genetic factors are. But because it's somewhat of a dual presentation of both Parkinsonisms and Alzheimer's disease, a lot of the studies have been focusing on Alzheimer's disease genes and Parkinson's disease genes to limited success. Some of these genes are, yes, also involved in Lewy body dementias, some others not really. It's only been more recently that this phenotype has been studied as sort of an independent disorder to discover new genes that are specific to the Lewy body dementias. But still a lot of studies to be done, and I'm excited to be involved in these kind of efforts together with Dr. Galvin. So what do we need to move forward? To be able to provide precision medicine to all patients, I'm using the same keywords as Dr. Galvin used before too, right? We need to be able to target treatment specifically to each individual, specific to what they have, what their background is, and so on. We need equitable representation of all the populations group in dementia research, especially genetics. Right now, most of the data is based on European patients, but that might not be as applicable to all the other groups. So when we develop treatments, we want it to be applicable to everybody, right? Or at least know whether it'd be applicable to the other groups as well. Just to iterate that this inequitable representation of the groups is not specific for dementia. This is actually a paper from a few years ago where they looked at all sort of medical research, and you see the big blue part, that's the European descent group, and all the other colors are all the other population groups. So there's a gross underrepresentation of those groups, right? So, but again, to be able to make sure that all the treatments are universally applicable to everybody, we need participation from all the groups. Okay. Alzheimer's disease is trying to do a good job. The research group in general has been really trying to fill in the gaps of the other population groups. Luckily, sort of, for frontotemporal dementia and dementia with Lewy bodies, because we're still in somewhat early stages of getting the genetic research done, we're still able to sort of prevent these bigger gaps from happening as long as we make an effort now to get bigger groups together. As I mentioned, we certainly have an issue because these two disorders are less common to get the bigger cohorts together, right? But eventually, we want to get to identifying all the genetic factors so we can understand what's happening in disease. And ideally, for all of these disorders, we'd like to include patients with a strong genetic contribution, so either having a positive family history or have an early onset case. That will help us understand what mutations are in those genes, and that will help understand the process for all patients. Okay, last slide. I have no idea how I'm doing on time, but last slide is sort of all the studies that we're doing at the HIHG, the Hussman Institute for Human Genomics. So we have several studies on Alzheimer's disease, studies for studying families with positive family history, patients with early onset case. We have specific studies for African Americans, for Hispanics, and myself am involved in all the related dementias to genetic research of frontotemporal dementia and the Lewy body dementias. Please write down our website and our phone number if you're interested. I'm going to be in the corner, I guess after the questions, I'll be the corner with the CCBH. Nothing prevents you from signing up for both studies. Just letting you know, you can sign up for Dr. Galvin's studies and for my studies. We do work together, so we will try our very, very hardest to not have you do things double. Our clinical phenotyping for the genetic study is less than what Dr. Galvin does, but he has very excellent phenotypic characterization of the patients, so we will definitely work with him for the Lewy body dementia studies. And I think that's it. So these are all the PIs at the HIHG on all the different genetic studies for dementias in University of Miami. I want to point out our study is in Miami, but you do not have to travel to Miami. We're more than welcome, more than happy to come to your house or do everything remotely as well. So don't be afraid of that. Thank you.